Y vamos a pasar ahora a la conferencia del doctor Pietro Ferrari de la Agencia Internacional de Investigación en Cáncer. El doctor Pietro Ferrari es jefe de la Rama de Metabolismo y Nutrición de la Agencia Internacional de Investigación sobre Cáncer, también llamada IARC, en Lyon, Francia. Es coinvestigador principal en el estudio del European Prospective Investigation into Cancer and Nutrition, y su investigación abarca aspectos relacionados con la epidemiología nutricional, incluyendo la coordinación de estudios epidemiológicos multicéntricos y la evaluación de asociaciones entre factores dietéticos y de estilo de vida con el riesgo de cáncer. Ha trabajado en el desarrollo de metodologías específicas para el análisis de conjuntos complejos de datos que integran información sobre el estilo de vida y datos moleculares aplicados a la investigación de la etiología del cáncer. Agradecemos al doctor por su participación y él nos acompaña de manera virtual. Adelante, doctor.
Yes, buenas tardes. I believe you can hear me. All right. I think it's fine. Thank you. Yes, sorry. I couldn't be there, and then I apologize because I'm Italian and then I should be able to speak Spanish, but I'm not. So I will deliver this talk in Spanish. Thanks a lot to the organizers for the invitation. So I am briefly, this is the title of my talk. What I will do, I will introduce to the context of nutritional cancer epidemiology and tell you a little bit about some of the activities we're doing within the nutrition and metabolism branch, with few examples on ultra-processed food, plant-based diet, and alcohol intake. And then I will finish up with some concluding remarks. Cancer. I probably don't need to spend too much time here. This is a cancer incidence in the world. I love maps. These maps is good. It shows the incidence of cancer, overall cancer, in the world. I think it provides a general image of how cancer is heterogeneously spread in different continents. You could see also this is the situation in Europe and in some other countries. You could see that in several countries cancer has been increasing in the last 20-30 years. You can see in places where, like China, where cancer was low, you can see that it's ramping up very rapidly. And in France has been stable over the last 20 years. So the question is, what is the role of diet on the risk of developing cancer? So I think it's good to put everything in perspective. And so how widespread is cancer? Well, in 2022, I think these are numbers that we tend to forget, but approximately 20 million new cancer cases were diagnosed, and about 10 million people died from cancer worldwide. These are general estimates. And the bad news about it is that in 25 years from now, the 20 million are probably expected to become 35 million cancer cases. I mean, for different reasons, including the population aging. The good news is that the cancer mortality is going down for various reasons. Unfortunately, not so much for progress in cancer prevention, but mostly because of huge progress in cancer treatment. So the question is diet, are diet and nutrition modulating cancer risk? And then what is the proportion of cancer attributable to diet? So the first estimates, they go back 45 years now. So this is seminal work by Richard Doll and Richard Pietro, the two Richards, and they estimated in 81 for the causes of cancer. So you could see smoking 30%, diet 35%. You could see how uncertain they estimate. Their estimate was from 10 to 70. So a lot of unknown around this estimate. But still, they said 35% of cancer is possibly due to diet. Alcohol, 3%. And then they didn't necessarily have firm estimates on obesity and physical activity. Now, this is estimates produced a couple of years ago in the United States. You could see the smoking is 20%. It's a bit lower than what expected. Diet went down to 4%. Alcohol's 5%. Obesity 8%, and physical inactivity 3%. I mean, if you take a look at this, has been an exercise that went on in several countries, and basically estimates are not incredibly different. And the second thing, which is striking, is that if you sum up all these numbers, there is a 40%. Okay, and 40% is basically a kind of every cancer epidemiology that works in cancer prevention. We basically are confronted to these estimates. So the proportion of preventable cancer cases is 40%. It is incredibly difficult to go beyond that. Nutritional epidemiology, the context is incredibly complicated. We work with diet, of course. This is what we eat, but then foods, they come with nutritional content that is incredibly important because they give us information about the biology of food and how they interact with their host factor. There are factors related to what we drink, of course, alcohol, but also to our lifestyle. Tobacco, obesity is a new epidemic in several parts of the world, and of course, physical activity has been well. Some people they really think that physical activity is the one. I mean, apart from smoking, of course, but it is the number one suggestion that for cancer prevention that one could give to the general population. The environment, of course, is incredibly important. And many different facets of that, and there is growing and if not established evidence that metabolic health plays a role in this context. So I probably don't need to spend a lot of time to persuade you that nutrition is incredibly complex. It's difficult to isolate components because we do not tend to do one single thing or eat one single thing. There is a lot of issues related to exposure classification. When it's down to behavioral sciences, confounding and selection bias are there, and then we need to deal with that. We have identified associations between dietary components and cancer risk, but very often we do not understand the mechanisms, basically why we observe these associations and why certain dietary components or lifestyle components, they are carcinogenic. There is, of course, a lot of endpoint heterogeneity in cancer, unlike other non-communicable diseases, and very often we deal with small effect. So what are the perspectives? Shall we give up investigating the link between diet and cancer risk? I ask myself this question probably every day, and I still found good reasons to do that, but I think the answer is not necessarily obvious. We know that diets contribute to cancer risk. A lot of it is probably through obesity and metabolic dysfunction, but there are very many aspects of diet that we can think they influence cancer risk, but it's so incredibly complicated to understand them and investigate them in observational studies. So what can we do here? Well, there have been many significant recent technical developments that made it possible to integrate several types of exposure assessments into observational epidemiology. And I'm talking primarily about the availability of molecular measurements. So GWAS, this goes back 20 years, but then there are new acquisition techniques to generate molecular data. I'm thinking about proteomics, metabolomics, epigenetics data. And there have been also progresses in the analytical frameworks to assess causality. So is it very important that we use any weapons, is a bad word, apologies, we have to investigate these relationships in a very challenging domain. Triangulate evidence is very, very important. So IARC is the International Agency for Research on Cancer. It's a WHO cancer agency. We have a mission, and it's basically the first one is to describe the occurrence of cancer. The map I show in my second slide is coming from one of the branches here. We understand the causes of cancer. We try to understand the causes of cancer, and this is where my branch is pretty busy with. And then we also implement preventive strategies. And in general, what we do is we promote global collaborations among researchers, hopefully for reasons that will become clear in the course of my presentation. So I want to spend probably 1 minute describing the nutrition and metabolism branch that I'm leading. So this is a multidisciplinary environment. We have expertise in cancer epidemiologies, in biostatistics, and also in laboratory facilities. And why is that? Well, basically because one of the domain, or one of the studies that we coordinate is the European Prospective Investigation into Cancer and Nutrition. Probably I don't need to spend a lot of time describing this, but this was a multicentric European study that was initiated back in the 90s that involved 10 countries and 23 administrative centers, as you can see in the map. And one excellent idea that researchers had back then was to collect biological samples that is currently still stored in liquid nitrogen at IARC. So this is a little bit the design of the study. You could see it started in the 90s. Dietary and lifestyle questionnaires were collected, blood samples was collected, and then there were follow-up questionnaires that made available some longitudinal data to be used in etiological analysis. Cancer ascertainment is happening through follow-up. I don't know about Mexico, but Europe, we are experiencing a very huge problem about data protection. GDPR regulation by the European Commission has made our life incredibly complicated, particularly for scientists that would like to work in international context. So the large amount of biological data, of course, justify the investment into laboratory sciences. So my colleagues Sabina Rinaldi, Pekka Keski-Rakkonen, and Chrysovalantou Katsoyannis, they are leading laboratories within the branch. And you could see that there are various techniques going from the quantification of targeted assays for hormones and inflammatory markers and fatty acids to the left-hand side of the slide, and moving to the right, we go more into untargeted applications, and so for measuring polyphenols together with targeted proteomics through the Olink platform, and going down to the liquid chromatography mass spectrometry to measure untargeted metabolomics. Of course, this is a variety of techniques that render possible the generation of an increasing amount of molecular data that is used in our study. So this is a little bit our context. So we have a set of exposures in the black, in the yellow box, that we can relate to the disease outcomes in the gray box. And then we could also appreciate or estimate or evaluate the role of, well, you call them either modifiers or mediators, very often molecular data. And then these analytical frameworks has been used to investigate kind of mechanisms. And so we have a lot of dietary factors, of course, as well as lifestyle factors and biomarkers measurements by means of metabolomics, proteomics, epigenetics. We also try to have research on microbiome. It's not obvious because we don't have fecal samples, at least in EPIC. And of course, our interest focuses on several cancer sites, but also cardiovascular disease, type 2 diabetes, and other non-communicable disease. All-cause mortality is also in our agenda. So I want to give you two or three examples of studies that we conduct. So using the same analytical framework, this is a study on healthy plant-based diet. So last year, Reynalda Cordova with Heinz Freisling here in the picture, they have found an inverse association between a healthy plant-based diet and cancer, type 2 diabetes, cardiovascular disease, and multimorbidity. This was a very fine study, and we're quite happy. But then we thought, okay, can we understand a little bit what is the biological pathway linking healthy plant-based diet with diseases? And so we used proteomics, and Peggy, Lair, and Heinz are working on a fairly complex analysis that I'll be happy to guide you through. So the healthy plant-based diet is a diet characterized by a large amount of vegetables, fruits, and legumes, of course. And then we had proteomics data that could inform or potential links on these relationships, because they could inform about a number of biological processes within our body. So this is going to be probably a little bit technical, but I try to guide you through that. So we have within EPIC, in a case cohort sample, about 12,000 studies participants with the 7K SomaScan proteomics data. Basically, there are 7,000 aptamers that inform about proteins available in 12,000 participants. And then we run relatively complex analysis. So in the first step, we related the proteomics data to the healthy plant-based diet. And then we identify signatures of plant-based diet. And the signature was made of 1,200 proteins. And of course, this was a big number. So we thought, okay, how can we screen them? So we used the CISPQTL, Decode Genetics, which is basically an online catalog. Well, basically what they do is we use genetic information that was strongly associated to proteins, and just what we want is to screen out those proteins that had strong instrumental variables, so that they showed a strong link with the proteins. And of course, we lost about 2/3 of the proteins because for the majority of them, we didn't have a strong enough instrumental variable, because our final objective was to run a Mendelian randomization analysis, whereby we could use genetic data that are available to relate proteins to specific disease outcomes. So we were left with 67 proteins. The next step was to run colocalization, and then eventually we could identify 9 proteins. Colocalization is a type of analysis whereby we have a look at the genetic region that is related both to the disease of interest and to the protein of interest. And if that the two are the same, we can say that the specific SNP, that the protein and disease, they co-localize if they have a common genetic origin. And so this is a strong evidence for causality. And so among the different and the 9 proteins, I'd like to focus on 3 of them. The CCM2 protein was associated to colorectal cancer risk. And then, of course, this makes sense because this protein is responsible for angiogenesis, is oxidative stress, and also is involved in inflammation processes. The NCAN type 2 diabetes link also made sense because the NCAN protein is a marker of metabolic dysfunction and is associated to steatotic liver disease. And this last one is a PCSK9 that was associated to cardiovascular disease. Well, the PCSK9 is a protein involved in LDL metabolism. And this is even a target for potential pharmacological studies. So this is ongoing studies, but that I think what I'm trying to show here that we are using some of the newly generated molecular data to inform on diet-disease link and see whether we could identify candidate and sensible molecular pathways that could further be investigated. I move now on to the second example is ultra-processed food and cancers. This is a study that was led by my colleague Inge Übrichts, and basically relating the diets following the NOVA classification of food processing was linked to the risk of cancer. So basically, participants were ranked into minimally processed food, NOVA 1, some processing NOVA 2, processed NOVA 3, and ultra processed food NOVA 4. And there were substitution analysis that were implemented. So what we found, apologies if this was a little bit busy, but on the left you can see different cancer sites and then associations between the 4 NOVA classification and cancer risk. And you could see that there were several inverse associations for NOVA 1, suggesting that a diet that doesn't make use of food processing was inversely associated to risk of several cancers, whereby the NOVA 3 we observe several associations for NOVA 3 diets and several cancer sites. And then the NOVA 4 was associated to head and neck cancer. Of course, we were quite surprised to observe this association, particularly NOVA 4 and head and neck cancer. And what we did next is Inge and Heinz, they worked on a paper that was published last year. No, actually, this was back in 2023. But we were relating the ultra-processed food to cancer, to risk of cardiovascular disease and type 2 diabetes. So what I want to suggest here, show you here, is that positive associations were observed for the 3 disease outcomes. And this is the black square. But then when they adjusted for BMI, therefore checking how much of this association was going through the obesity pathway, you could see that there was very minimal change in the risk estimate for cancer, some change for cardiovascular disease, and of course larger change for type 2 diabetes, because obesity is such an important risk factor for obesity. These studies, at least for cancer, suggest that the adipositive hypothesis on why ultra-processed food might be associated to some specific cancer is possibly quite weak and could at best explain 20 or 25% of the whole relationship. That's why we have a study, and we develop an application that was submitted to the World Cancer Research Fund. And this is a study where we are hoping to investigate the candidate mechanism of carcinogenesis in the associations between ultra-processed food and risk of breast and colorectal cancer. And this is something that we developed together with Martin back in February, and now we are awaiting a response from WCRF. Alcohol, this is again another map. This is the proportion of cancer attributable to alcohol in different parts of the world. Of course, this reflects largely different prevalence of alcohol in different parts of the world. There is another heterogeneity element, which is alcohol doesn't necessarily influence all cancer sites the same way. Alcohol was associated to cancer of the upper aerodigestive tract, female breast, colorectal cancer and liver cancer. But then there were questions whether there were associations with less established cancer sites. So 10 years ago now, I started a large collaboration with Dr. Stephanie Smith-Water at the Harvard T-Chan School of Public Health. And we were using resources of Diet and Cancer Pooling Project that she coordinates at Harvard. And so we focus on cancer sites that have an inconsistent prior evidence with alcohol intake. And at IARC, we focus on upper aerodigestive tract cancer in never smokers, pancreatic cancer, and thyroid cancer. So you could see here, these are all studies that we just published. We were able to confirm an associations between alcohol and UADT cancer in never smokers. Actually, the associations were very similar in never smokers and smokers. We found a positive association in pancreatic cancer, and also an inverse associations for thyroid cancer. I'm showing women here, but it was very similar in men. We have not necessarily great ideas on why there is an inverse association. And the inverse association is also confirmed for kidney cancer and non-Hodgkin lymphoma. We try to expand a little bit this work and see whether we could identify a biomarker of alcohol. And this was a collaboration that we established with colleagues at NCI. So what we did was we used untargeted metabolomics with a fairly complicated multi-tier design in EPIC and the ATBC cohort. And we could identify actually two features that were associated to alcohol intake. You could see the dose response curve of one of them. So one feature, we couldn't annotate that back then. This was 3 years ago. But the first one was the 2-hydroxy-3-methylbutyric acid. And then the good thing about this, well, I don't know whether it was a good thing, but the interesting feature of interesting element of this study was that unlike alcohol intake, this feature was strongly associated with hepatocellular carcinoma in EPIC, pancreatic cancer, as well as liver cancer in the ATBC study. And this was quite surprising. So this feature has been observed as associated to alcohol in previous cohorts before. And I need to spend 30 seconds to say this. So this study started as a biomarker discovery study, but then we realized that probably what we have identified was not necessarily a biomarker of alcohol intake. It was more something that was informing us about the metabolism of alcohol intake and possibly potential mechanism, because the 2-hydroxy-3-methylbutyric acids is linked to rate of hepatic glutathione synthesis that it can increase in response to oxidative stress. And oxidative stress, in fact, is one of the candidate mechanisms for alcohol carcinogenesis. So what we did that, and this is my last slide before the concluding remarks, is that we try to validate these in intervention studies. The women alcohol study that is led by colleagues at NCI. And so they enrolled 51 women. There was a 8-week treatment periods with a crossover design, and that women were given different doses of ethanol, well, never exceeding 30 grams per day. So we confirmed the results of the 2-hydroxy-3-methylbutyric acids, and also 45 other metabolites were identified. Ethyl glucuronide is quite well-known biomarker, but also the ethyl alpha glucopyranoside, they showed very strong association with alcohol intake. So now the ball is back here, because with the data we have and the nested case control study, we could check the associations between these features and cancer risk. Yes, I would go to the concluding remarks here. Is nutritional epidemiology generated quite a lot of evidence on the role of diet and nutrition in cancer development, but this evidence is not necessarily always replicated. And if we take a look at the table from the WCRF of the different dietary exposures and different cancer sites, in fact, the table has many different gaps, and I think it would be good to know more about all these gaps. That there are incredibly new opportunity provided by omics data, both for biomarker discovery, but particularly for identification of biological pathways. Of course, the need of replicate findings, triangulate findings, never trust your results, always try to replicate, if you can, also with intervention studies. I don't think what we did was correct. Usually what you want to do is do an intervention study first and then replicate the findings in an observational setting. We did the other way around, but it is what it is. I didn't talk about that, but there is a great need to translate what we find. But there is also a lot of input. I loved Martin asking Abraham about, hey, you are the new generation. We want to generate leaders. I think leaders need to have rigor and be creative. This is the secret, I think. And of course, collaborating with smart people is essential. You can find the references of some of the things I told you today in a paper that was published in April in JNCI monograph. I need to thank a few people, Elmira, Diana, Peggy. They were busy with some of the analysis here. The colleagues in the nutrition and metabolism branch, of course. I don't know whether Isabelle Ormieux is there, but anyway, she was heading this branch 10 years ago, and we are still thankful to her contribution. A lot of good people in EPI, colleagues at NCI, and of course in the Diet and Cancer Pooling Project. And with that, I thank you for your attention.
Muchas gracias al doctor Pietro por su presentación. Fue sin duda muy interesante. Y con esto nos acercamos ya al cierre de nuestro evento conmemorativo. Para